gene sequences from NCBI. First, we have to go to Google search engine. In that search engine box, we have to type NCBI. Once we typed NCBI, we can get the NCBI Center, that is National Center for Biotechnology Information. Under this we could see BLAST, nucleotide, PubMed, protein. When we are selecting this National Center for Biotechnology, we will go to the main page of NCBI. In that main page, we can see different categories, that is all resources, from where we can take all resources, and the chemicals and bioassays, all details also we can take, data and software, DNA and RNA, domains and structures, genes and expressions. All these categories we can see at one side. And in the main part of this NCBI, we can see submit. So this submit means when we are sequenced the DNA of any organisms, we can submit here. Download, learn, develop, analyze and research. All these will be given here. Now we are going to retrieve nucleotide sequences or gene sequences. That is why we are selecting from this all bases. Under this down arrow, we can see different categories. Of these we have to select nucleotide. Now type the protein, its sequences we are going to retrieve. I am selecting ATB4. Select search. You can see this is human breast cancer tissue. It is taken from human breast cancer tissue and it contains 286 base pair. This is the mRNA sequence and its accession number is this one. This is the accession number. Accession number means once we are sequenced the protein or DNA, when we are submitting it in the NCBI, they will allot one number, that is what known as accession number. The sample is taken from Homo sapien, that is human. Authors, this is submitted by Brickett, Cho, Gao, all these are authors who submitted these sequences. And they have given the title like high throughput cloning of full length human complementary DNAs directly from complementary DNA libraries optimized for large and rare transcripts. All these details were given here. At the last, it is the gene sequences. Here it is given 161, 121, 181, 241. This means in the first row the 60 sequences are given. The second row starting from 61 61st sequence, and it is extended up to 120 sequences. So each row contains 60 sequences and it is in the form of 6 blocks. So each block contains 10 sequences, you could see here. Now we can copy these sequences like this, or otherwise we will get it in the form of FASTA format. The FASTA format, when you are clicking this, selecting this FASTA, you will get FASTA format. How can we identify the FASTA format? By this symbol. FASTA format always start with greater than symbol. So here all the letters will be given in capital form, and this means GC, that is guanine, cytosine, adenine, cytosine, it will be given like that. Now we can copy this and we should open the notepad. There we have to paste. Why we are selecting the notepad? If we are copy and paste in the ordinary Word file, we cannot use it further. That is, when we are comparing two sequences, we cannot use it further. So that is why we should paste it in the notepad. This is how we have to retrieve the gene sequences. Now I am saving the file as ETB4 sequence. Hope you understood how to retrieve the sequences from the NCBI.